Welcome you all to a new video of Sunanda's tutorial. So it is the part 2 video of protein sorting, in which we will discuss about mechanism of a particular type of targeting, that is co-translational targeting, CTT. So what is protein targeting? It is a cellular mechanism or intracellular trafficking that delivers the newly synthesized proteins to its specific destination. It can deliver the newly synthesized protein into plasma membrane, nucleus, mitochondria, Golgi complex, lysosome. So this process is very important for cell homeostasis, with dysfunction often leading to disease. When dysfunction takes place in protein sorting and trafficking, it can be lead to a serious disease. Okay. Now let's discuss about the mechanism of a particular type of targeting, that is CTT or co-translational targeting. So here protein is transferred to its destination during the translation, most commonly to endoplasmic reticulum. So here a signal recognition particle binds to emerging hydrophobic N-terminal signal sequence. So generally the proteins, newly synthesized proteins, have a signal sequence which can detect the newly synthesized protein and its destination. It's like a pin code. Signal sequence is like a pin code which can detect the address of that particular protein. So SRP or signal recognition particle first attach to that signal sequence of nascent polypeptide or freshly manufactured polypeptide protein linear chain. It leads to the pause of translation, guiding the ribosome nascent chain complex to SRP receptor on the membrane or endoplasmic reticulum membrane. So what are the key steps? At first, recognition. As polypeptide chain or linear chain of protein emerges from ribosome, newly synthesized protein emerges from ribosome, SRP. What is SRP? Signal recognition particle binds to hydrophobic signal sequence, because newly synthesized proteins have a signal sequence and SRP binds to it. Then SRP ribosome complex moves to target membrane, where SRP binds to the SRP receptor. SRP receptor is present on the surface of ER or endoplasmic reticulum. It is ready to receive the SRP complex protein. Okay. Then transfer through a GTP driven mechanism. Ribosome complex is transferred to translocon channel, for example Sex61 on the membrane. Then translocation, SRP releases ribosome and returns to the cytoplasm. Then translation resumes, forcing the nascent polypeptide through the translocon or inserting into the membrane. Then finally the signal peptidase clips off the signal sequence, because it is not required further upon entry into the ER lumen. So at first, ribosome synthesize a protein with signal sequence, then SRP binds to it, then they all transfer to the ER membrane. On ER membrane, GTP binds to it, and this attachment leads to the hydrolysis of GTP, forming GDP, and finally these protein enter into a translocon channel, and SRP signal sequence both detach from that protein. So this is the whole process. At first, targeting, which is SRP dependent, then membrane docking, that means attachment of that complex to ER membrane, then GTP driven transfer. Both SRP and SRP receptor are GTP binding protein. They bind with each other it induces the hydrolysis, that means breaking of GTP to GDP. Guanosine triphosphate break to form GDP or guanosine diphosphate. Then ribosome translocon engagement, this hydrolysis drives the dissociation of SRP and SR complex, releasing ribosome, allowing it to bind with the Sec61 channel, which is a translocon channel. Then translocation takes place, that means protein gets inserted inside the ER lumen, okay, or the translocation channel. So this is the whole process of your co-translational mechanism, okay. Then after this type of transportation, the translation resumes, okay. The growing polypeptide is directed through the translocon into the ER lumen, and again the translation resumes, restarts. So it is the whole process of CTT or co-translational targeting. In my next video, I will discuss about post-translational mechanism. So please stay tuned and please subscribe to my channel Sunanda's tutorial. Thank you.